Recording.
So good afternoon.
Yeah, good afternoon.
Good afternoon, sir.
Yeah,
So am I audible,
good
sir?
afternoon.
My voice is clear or not?
Yeah, your voice is clear.
Yeah. Thank you so much, sir. So hello everyone. I am Mr. Yasser Kazi, the coordinator for today's session. So on behalf of YP Shaman College of Pharmacy Institutions Innovation Council, I welcome all the participants in this Impact Lecture Series, Series 2 of Series 2. Okay. So this actually, as you know, that we had initiated this Impact Lecture sessions from the last year, and once again we are coming here to conduct the Series 2. So today, in the first session of the Series 2, we are having two important topics. The topic number 1 is research and development in pharmaceutical formulations, and topic number 2 here is IPR in pharmacy current scenario. So for this, I may request Principal Abu Bakr sir to share some few words.
Yeah, good afternoon, everyone. On behalf of management and chairman, I welcome Mr. Irfan Ahmed Farooqi, Scientific Project Manager,
Thank you.
Formulation and Development, Inventia Healthcare Private Limited, Thane, Mumbai. As well as I welcome all the delegates who have joined this Impact Lecture Series under the IIC. I request Mr. Yasser Kazi to continue with the lecture.
Thank you, sir. So here I would like to introduce today's eminent speaker, Mr. Irfan Ahmed Faruqi sir, Scientific Project Manager, Formulation and Development, Inventia Healthcare Private Limited, Thane. Sir is having 17 years of experience in formulation research and development. He is leading and guiding team of scientists for the past 10 years. He is having expertise in F&D and NDDS, and he has worked as a research scientist in Jubilant Life Sciences, research associate in Alchem Laboratories, and group leader in IPR Laboratories. So, without wasting any time, sir, Irfan sir, I would like to request you to initiate this session.
Yeah, thank you, Yasir sir, and thanks
Thank you,
Babaji sir as well. So, as he already introduced the today's topic, I will be giving the discussion or the some overview for the formulation development, how it is being done in the industry. So it will be just a glance for the different ideas. I'm just sharing my presentation.
अरे वेबिनार अटेंड नहीं कर रही है आज का? करो ना, एक घंटा का तो है वो। हाँ,
Yeah, is the slides visible?
Yes, slides are visible.
Okay. So as the topic is clear to you all, and I'm just proceeding for this presentation. But before I talk further, just put me a disclaimer that whatever I'm going to present, these are solely my views on this topic and not of the Inventia Healthcare Limited where I'm working currently. So coming to the topic, we regularly talk about the formulation development, and if you may be basically aware, so formulation development is nothing but a combination of drug substance and excipient which can be presented in a suitable dosage form for the ease of administration and the patient comfortable. So basically whatever drug substance or the pharmaceutical ingredients, the active pharmaceutical ingredient is present or the combination thereof. Am I audible?
Yes, sir. Yes, sir. The voice
Yeah.
coming.
Okay. So the active pharmaceutical ingredient or the drug substance, they are never presented in the pure form for the administration purpose, and always they are accompanied with some set of excipient depending on the dosage form what they has been incorporated into. But exactly when does it start? The formulation development normally starts once the clinical phase 1 is almost over. It can and there from the candidate drug is selected, and that can be presented as a medicine for the future evaluation purpose and the patients. So what are the factors that goes into consideration of the pre-formulation or the formulation development? I will request for the audience, if they want to question in between, they can question if they are not clear, or they can put the questions after the end of the session. Both ways they are welcome. So whenever there is a drug which gets shortlisted during the clinical, after the clinical trial, then basically what we look for is to provide this medicine in a suitable form that should be effective and usable. At the same time, it should be stable for a certain period of time, which we call it the shelf life. And majority of the patient should feel it comfortable, and it should be acceptable to the most of the consumers. So that is the basic minimum objectives for this formulation development. So how the choice of formulation is derived. So basically there are various aspects into this development. First is the pre-formulation. Pre-formulation studies, which are basic, trying to understand the physicochemical nature of the drug. And then there will be analytical method development for the evaluation of this drug. And the next step will be excipient screening. And finally, the medicine will be presented as a suitable dosage form. So basically, the pre-formulation is a very broad perspective, but what we look in the pre-formulation studies, I'm just have put here the few points, which is the physical state of the API which has been shortlisted, whether it is solid or liquid, and understanding about this crystal nature or the polymorphism of this API, solubility of the API and particle size, shapes, and surface area, and then pH and pKa values. So these are the physical and chemical attributes of any API, which helps us in determining what will be the most suitable approach. See, solubility is one of the most important factor which plays a critical role in delivering any dosage form to the patient. Whereas we say its solubility, it is related to evaluation in terms of PCS classification as well. There is a system called BCS classification where we intend to have a solubility of the drug in the water with a limited volume, that is approximately considered as 250 ml. So where we try to see the solubility of API, and since the in vivo system is dynamic and the GI tract, if we are administering, or even the plasma system, these are all having a certain pH values. So it is preferred always to check the solubility of this API across all this pH range. So this will help the scientists to determine in which form you want to present this drug and how much will be the support required from the other excipient or the ingredient. Similarly, polymorphism is the existence of the drug substance in a different crystalline structure. And you may be aware, some of you may be aware that all polymorphic forms are not active. Some are inactive or they will be lesser active as compared to the original molecule. So there may be no polymorphism at all, and there may be sometimes two polymorphic form available, or it may be more. But eventually there will be only one polymorphic form which will be more effective for the best pharmacological effect. So likewise, particle size is something which determines again as critical, which we can say is as critical as the solubility or the physical state. Particle size, normally the scientists try to evaluate the performance of the drug with the minimum bare minimum particle size as much as possible in the in the physical form existence, and there they check the performance like flow properties, bulk densities, and like static charge generation, and how that with the increased surface area, what will be the impact on the solubility of the drug substance. At the same time, the particle size is directly going to affect the flow properties of the molecule. Similarly, pH is also a critical the point, because the pH will determine the ionization of the drug substance, and ionization ultimately determine the absorption. So after going through all this pre-formulation study, there will be the selection of the dosage form, ultimate dosage form, in which one intend to present this drug substance for the patient or consumer's use. So grossly, dosage form can be defined by various mechanisms, but roughly we can present this dosage form as an enteral formulation, parenteral formulation, and for the topical. So again, the dose, when it gets selected in the preclinical and clinical phase 1 study, then based on the solubility and effectiveness of the medicine and intended function, one of the route will be selected as a delivery system for the patient, whether it will be a oral route, solid form, or parenteral route like injectable or then ophthalmic solution or ointment cream, topical, etc. So after understanding and deciding the which dosage form to be selected and what are the physiochemical characteristics of the API, then comes the time for the selection of the various excipients. These excipients will be selected based on the dosage form and the intended support required by the drug substance to be delivered. So for majority of the dosage form, there will be one diluent or a vehicle which will be needed to deliver this drug substance. Ideally, the diluent is expected to be most inert and non-reactive towards the drug substance. And this compatibility will get determined during the pre-formulation studies for these drug substances. Similarly, if it is a solid oral dosage form or the capsules or some other delivery system, there may be need of some binders which can support to deliver this medicine in a particular design form and keep it adhered to the other excipients which are present in the system. And if it's a solid oral form, then there will be requirement of disintegrating agent, and whether it is a suspension or emulsion, then accordingly there will be need for suspending agent or emulsifying agent. pH modulators are a must for the solution, suspension, or basically liquid delivery dosage forms. And sometimes there will be certain drugs which will be required to have some stabilizer or additives like preservative and antioxidant to enhance the stability of the drug substance. So these are the basic startup for the development of a dosage form. So ultimately, what are the dosage form? If we go into the details, so there are the things which we regularly use like tablet and capsules, which are normally administered twice or thrice daily. So in terms of pharma industry, they are called as immediate release formulations. They can be tablet, oral, capsules, oral solution suspensions, or it may be ointment cream, which can be applied topically. And there will be some injectable that will be for the intravenous route. So these are the dosage form which we are routinely accustomed to use, and these are the most suitable and widely used dosage form. But the limitation with this conventional dosage form is, as you may be aware, there will be repetition of the administration for the longer therapy. So this becomes sometimes irritating and mostly results in non-compliance of the patient. And there are certain drugs which are required for the lifestyle management. So those are the drugs which are required to be administered for a longer duration of the time, and they are normally administered for the years. So these are the drug substances or the active ingredients where we need to have some slightly different dosage form, which normally are termed as non-conventional dosage form. But this is just a relative term. You can term it in a different way as well. So normally under non-conventional, we classify novel drug delivery system, transdermal devices or patches, inhalers, metered dose system like aerosol. Then comes the suspension in emulsions with the nanoparticles or the liposomes in their system. And there are the new class which is coming into the market, that is a complex drug products, or what you call as a biosimilar products or the complex drug substances. So we'll just have a look through this one by one. The novel drug delivery system, we can classify a few of these kind of dosage form like extended or prolonged release tablets and capsules. There could be osmotic drug delivery systems, then multi-particular system, and delayed release system. And by delayed release system, I just don't mean that it is just entry coated formulation. This could be having a delay of more. For the delayed release or entry coated polymer, it is expected to have a drug release be after 2 hours, which is normally adopted for the drug substances which are having either which shows fast pass metabolism or there is instability in the acidic environment and you want to pass the stomach. Otherwise, there will be certain drugs where we want to have a targeted delivery, like in the later phase of intestine, or the colon part. So that kind of formulations are also classified under the delayed release system, because the onset of the release will not happen immediately. That will take a longer time. And as the drug passes through the GI tract, then this coating or the membrane through which the drug release is controlled, it will gradually open up and then deliver the drug at the end of the intestine or where the target is. Mostly these kind of systems are adopted for the drug release beyond pH 5 or 6. So there are different technologies adopted for this achieving this kind of deliveries. And opposite of that one is the mouth dissolving strips, films, or sublingual tablets, or immediately more dissolving tablets as well. So the here it is the opposite, where the onset of action is intended to have very quick, and we want to achieve the drug effectiveness in patients who are facing vomiting or some sort of gastrointestinal discomfort. There also this kind of more dissolving systems can be utilized. So one of few example is the nitroglycerin strips, which is normally used for the ischemic heart patients, and hyoscyamin, which is used for the abdominal discomfort or the bowel discomfort. Similarly, antiemetic drug like ondansetron also used delivered as a strip, soluble strips, which we have to just place in the mouth and it will get dissolved within seconds. So coming back to this extended release or the osmotic delivery systems, osmotic drug delivery systems are normally meant when we want to have a longer drug release profile and we want to achieve a zero order. Here again, there are different mechanisms which are adopted to achieve this osmotic drug delivery system. So based on this formulation type, what we are selecting, then accordingly the role of excipient will change, and in compliance or in compliance or considering the drug substance, we have to select such an excipient which will help us in achieving this intended drug delivery. For example, if we want to achieve extended or prolonged release, then we may adopt for insoluble or soluble polymers which have the high viscosity and they can hold on to the drug for a longer duration in the after administration. And at the same time, this can deliver the drug at the constant rate. Similarly, multiparticular systems are the drugs where we want to deliver it through we want to hold the drug release for a certain period and then suddenly we want to release the drugs after a certain time. The added advantage for this multi-particular system is, though it is not very well used in the India, but in other countries, the administration is enhanced by using the apple sauce or some kind of juices where these pellets are sprinkled and taken along with as a suitable dose. This sprinkling allows that patient to have a quick solubility, because for the older patient or the smaller kids, swallowing something is a big challenge and they don't prefer to take the medicine because of this reason. So likewise, there are the transdermal devices or the patches. Again, there is a lot of set of excipients which we need to look into this if you want to go for the transdermal devices or patches. And normally these are the drugs where it is intended to have a longer duration of release and without the discomfort of taking this medicine through either injection or through oral route, which we want to avoid. And this patch or the device will be applied to the skin part, which will not be visible to the other person. And over a period of time, this drug will be released through the skin. So there are again different approaches for this one, where different kind of device or the patches will be developed. But this kind of dosage form has the limitation. Mostly we can deliver with this route only the drugs which are more potent and which require lesser amount of dose. The dose which are very high is difficult to deliver, because then you have to prepare a patch which will be too bulky for the application purpose, and again that will cause the discomfort to the patient. So mostly hormones are the favorite and the contraceptive, which are normally presented by the transdermal patches or the devices. Then there is one more class called inhalers and aerosols. These are the system where the drug is targeted for the lung or respiratory tract system, or we want to have the absorption through the nasal route or in the mouth itself. So mostly for the asthma, this is the preferred route of administration. There are nasal drops which are nowadays delivered through the nasal mist. But this kind of devices, actually this kind of dosage form needs for special devices, and the construction of device is very critical for delivering the perfect dose, because every time it has to be the same amount of dose which needs to be delivered effectively to that patient. So when we come to this kind of formulation, again, the particle size is become the more critical aspect for any drug substance to achieve this kind of, to be suitable for this kind of delivery system, or at least that drug substance has to be soluble in the required amount of the solvent or delivery vehicle, which can be used as a mist form application or the drops. So the criticality of understanding going or having the pre-formulation knowledge of that drug substance is very evident for such kind of meter dosage forms where we want to have this kind of delivery. Again, this route of administration is favored only for the limited drugs, not for every drug, because the administration or whatever drug is administered will ultimately reach to the lung, or it absorption has to happen through the nasal tract, which offers a very smaller absorption area. Then there is a complex drug product, and why this has been termed as complex, because either the structure of this drug substance will be very complex. Maybe it will be a larger peptide or a great molecule, which is as compared to routinely administered drug will be very huge to characterize and define, or the device which is being used for the delivery of the drug will be complex. So there are various examples available for such kind of the drug product. So these are broadly classified into complex drug products or the generic product. So few of the examples are these two dosage form which I am mentioning here. One is the glucagon and another one is for the eye drop. So in both the cases, FDA has adopted a different strategy for the approval of these drug products. So basically, when we are running a formulation development and there will be entry of the branded or innovator company in the market, after all the studies and establishment of the effectiveness and the suitable dosage form, there will be the second set of the companies who will be competing against that innovator product, and they will try to market or launch the same drug substance post patent or by non-infringing that patent, and they will try to copy that formulation. So part of the pre-formulation activities, or you can say most of the activities, is done already by innovator company. Now they have to understand or deformulate this existing formula and then devise their own formulation to be bioequivalent with this available product in the market. But there are challenges for these complex drug products where establishing bioequivalence or the equal performance of the new developed product will be challenging, and this will be totally a different set of exercise apart from the routine activity which we normally perform for the most of the medicines where there is a simple bioequivalence study is done, or you have shown the passage of drug for in case of the transdermal patches, infusion of the drug substance through some membrane, and then we get the approval. So for this complex drug generic product, we need to take some extra or novel strategies. So then there will be some nanosuspensions or emulsions. These are the liposomal drug delivery system. See, there are the lot of drug candidates, almost close to 40%, which are totally insoluble. Or sometimes soluble drug also is getting delivered by this liposomal delivery system, where we are using solubilizer or the surfactants, where with the drug particle or the drug substance is micronized, or you can say nano-sized. In fact, where the particle size actually less than 1 micron, and then it gets suspended or embedded in the surfactant or the solubilizer, and these are then forced to form into nanoparticles, which are then delivered for having a drug action and the targeted entry. One of the such example is the Infugem. This has been developed by the Sun Pharma, and this is anti-cancer drug, which in the literature you can refer on the net also for this detailed understanding. So this is all about the development of this different kind of dosage form. So what happens when we are talking about this different kind of studies or the way how the product is developed or understood? But now with the changing scenarios and the changing dynamics of the market, and in fact some regulatory requirements are also are getting stricter. There is a concept for the QBD and a PAT. So these are two different things which I'm just summarizing up in front of you. So QBD is basically the systematic development approach for the drug product, whereas PAT is the process analytical tool where we try to do the online analysis of the drug product. So the concept for the QBD is nowadays whatever the development happens, there the drug product is understood and the impact of different excipient or combination of excipient is understood one after the other. The manufacturing process get optimized, which then get scaled up, and then the batch size gets scaled up to commercial size, and then it gets commercialized. But during all these activities from R&D, smaller scale batch to the commercial scale batch, the transition of product happens with various stages. However, the standard practice is not to challenge too many parameters at a time. So almost every parameter from the material to the process, these are certainly fixed in such a way that there won't be any deviation, and that drug manufacturing process has to follow that certain route. So the QBD is something where we try to understand the major impacts of the changes. Say, for example, the quality of input material is changing. We are using one polymer, say, for example, methyl cellulose, which is having a viscosity of 4000. But as a standard practice, it will not yield always a 4000 CPS viscosity whenever we want to use. It will having a certain range. So in QBD, we try to understand what will be the impact when this viscosity changes to 3000 CPS or when it changes to 5000 CPS. And this will be understood in context of the all other parameters which might get affected in presence of the HPMC. So there will be some other excipient which will be like, for example, magnesium stearate, which is most globally used lubricant material. So say, for example, in contrast to presence of magnesium stearate at a certain level, for example, 1 mg, 23 mg, and what will be the impact with the change in viscosity of HPMC. These are studied together, not alone. So basically, this QBD is something where we try to create a design space and understand the variation which by default or inherent variation of the process or the material which we introduce in this dosage form, and how they will be impacting the end point of our drug product. Ultimate aim is to have a drug product which will have the consistent target and within the set specification, even though this variation may come or it may not come. So that is how briefly what I can summarize the QBD. So it's just basically understanding the critical process parameters and critical material attributes and the impact of these changes on each other without affecting the drug product quality. Now, when it's a commercial manufacturing, the standard process is drug product manufacturing process is extended up to certain step, then it will get stopped and samples will be sent for the analysis, which is normally called for the in-process analysis. So there will be some certain defined steps, for example, 1, 2, 3. After this 1, 2, 3 step, there will be a stoppage. That particular content will be checked for the drug substance, and there will be go ahead for the next step. And after completing these 3 steps, there will be a finished drug product, which will be again tested, and then it will be released in the market based on the correct result. So the focus is on the process analytical tool to stop this offline analysis practice and bring this analysis on the online. So the online system is targeted to avoid these stoppages after step 1, 2, 3. And after completion of the 3rd step, product will be ready to be sold out in the market. That is the intention for the pad. Sorry. So for the pad or QPD, these are the two concepts which are relatively new, we can say, for the pharmaceutical industry. And adopting this is also a big challenge, because both these technologies or the concept requires a lot of input. And once this data set is ready, then this can be useful. But however, for once adopted successfully, both will yield up into a good product which is having a good drug product quality. At the same time, there will be reduced release time, and that will enhance our product availability in the market very fast. So these are the two combinations of the new concept which industry is targeting to adopt, but still it is not that fully successful or not at that grand scale. So with the changing scenarios and this development of this computer industries, so there is a creation of artificial intelligence. So pharmaceutical industry is looking at this aspect also. But the challenge here is pharmaceutical industries normally keep their things quite secretive. They don't share the information with each other. Though there is a lot of data generated day in, day out, but it will not be in the public domain or will not be available for the other pharmaceutical player. And this data is somewhat scattered, which the companies, different companies or the software giants are targeting to pitch their artificial intelligence into the pharmaceutical industry, where they have developed different kind of tool. So one of such tools are like LMS, which is learning management system, and CMS, content management system, and then precision medicines. So nowadays a lot of people are using this smart gadgets where we can have the heartbeat monitored with just a small watch or the band on our hand. Lot of things are available like glucose watch, uh, and there are certain things where we can have our heart rate monitored regularly. So these are the things which the companies in the advanced technology are trying to implement in their delivery, so that there will be the ease for the patient. But again, these are the technologies which are not fully adopted yet, but there is a lot of scope in this collaboration of these softwares and the pharmaceutical knowledge, which can help in the future for the ease of administration of the medicine. So it's like they have developed one contact lenses which are embedded with the nanoparticles, where we can have a direct measurement of the glucose level, and the diabetic patient it just have to wear it, and without any impact or without multiple precision, he will be having the idea of this glucose level, what is going on in the system. Similarly, there are delivery devices where the special drugs are delivered with quite ease. Like there is a glucagon similar polymer or the glucagon administration, which is normally used for the hypoglycemic patients. So it's very complex peptide, but which can be delivered very easily.
सर वो बात की सर वो ट्रेनिंग हो की उनके यार
So if the agglomeration of this artificial intelligence pharmaceutical happens, this will be the good for the future of our industry. This is sorry. So I'm just concluding here with the words that this artificial intelligence agglomeration with pharmaceutical knowledge is a long path ahead where we can see a lot of improvement in future. So if you are having any questions, you can ask here.
Thank you so much, sir, for this very informative session. Now the session is open
Thank
for
you.
questions. So dear students, please ask your questions. So is there any questions from students or faculty side?
I hope I was audible and
Yes, sir. Yes.
succeeded in delivering the concept. See, my intention was to only introduce briefly the overall scenario, what is going on in this pharmaceutical development, what we call formulation development routinely. So it's a very very broad domain, and I just touched upon the various aspects of this. So in fact, each and every topic is having their own depth, and that will call for their detailed lectures if we keep individually. So if there are some questions, student can ask now, or they can approach me later. It's not an issue.
Irfan sir.
Yet,
Irfan sir. What are the developments at the industrial level which are taking place regarding BCS class 4? BCS class 4 drug. Yes.
Yeah. See, BCS class 4 drugs are something which are very difficult to develop, and normally these are some things very challenging. So there are ways for enhancing our solubility. So like hot melt extrusion technology is there, where you are changing the polymorphic form of that drug substance. Or we have to study the solubility enhancement, like in the presence of surfactant, where this drug is going to be a as a most soluble candidate. Or just I put this example of liposomal drug delivery system. So for the insoluble drugs, there will be some preparation of such kind of suspension or emulsion for this kind of insoluble or class 4 drugs. But ultimately for this kind of drug substance, it's first of all, the most critical point is to understand the kinetics of that drug substance. Once you get that idea, then it will be more convenient and easier for someone to develop this kind of BCS class 4 drug. So basically there will be two challenges. One will be to increase the solubility, and second one is to enhance the bioavailability of the same system.
Thank you. Okay.
Yeah.
Any other questions? Please do ask your questions, MPharm students, BPharm students.
So yes, sir, can we conclude if there are no more questions?
Yes, sir. Sir, we are very thankful to you, sir. We came to know many things about formulations, conventional and non-conventional dosage forms, NADS and QBD and PAT. So very thankful, sir. So just to conclude, sir. Thank you so much.
I think we can conclude. I thank OIB Chauhan College of Pharmacy for giving me this opportunity to present my thought in front of your students. Thank you, Dr. Furqan and Dr. Bawazir sir and Yasir sir as well. Thank you. Thanks a lot.
Thank you so much.
Thank you. Bye.
On behalf of college, thank you very much, Irfan sir.
You are welcome, sir.
Okay. So we end this session here. Thank you once again, everyone.
Okay. Okay. Thank you.
So we are just waiting for the next speaker, Mangesh sir. I think we have to wait for 2 to 3 minutes. Okay. So dear participants, for our next lecture, I would like to introduce the next eminent speaker, Dr. Mangesh Jagannath Pawar sir, Assistant Director, Head IPR, Wokard Research Center, Aurangabad. Sir is having 22 years of experience in research and intellectual property. He is PhD from Pune University and he is LLB from Pune University only, and he is having MSc degree also. He has worked in different therapeutic areas such as anti-obesity, anti-diabetic, pain management, antibiotics, and anti-inflammatory. Sir has filed more than 30 patents and publications, and he is a qualified registered patent agent. So I would like to request Mangesh sir to continue with this session.
Okay.
Sir, please
Thank
continue.
you.
आपकी क्लास में लाइक तो नहीं है।
Good afternoon, everybody. I hope the screen is visible.
Yes, sir. Yes.
Okay. So today I'll be talking about intellectual property rights, and so we'll discuss the IPR in general. First of all, I would like to thank the organizing committee for inviting me to deliver a talk on IPR, especially to the Principal Dr. Bawazir, Dr. Lahoti, and Dr. Furkan Khan for coordinating and arranging the webinar. I'm equally thankful to Dr. Mahesh Patel, CSO, Wokard Research Center, for allowing me and encouraging me to participate in today's webinar. I'll be more happy if we can have an interactive session. Therefore, you can ask me any question without any hesitation during the webinar or after the webinar. Shall I start?
Yes, sir. Yes, sir.
Okay.
Yes, sir, you can start, sir.
Okay. Fine.
Dr. Mangesh,
Yes.
start.
Yes. As we know, there are different kinds of properties. Mainly, these are movable properties, immovable properties, and intellectual properties. It is very clear that movable properties we can move from one place to another place. Immovable properties that we cannot move from one place to another place. And there are intellectual property, which is a result of intellectual inputs. It is mostly related to inventions and literary works. Now we'll see the nature of intellectual property. It is, as the name itself indicates, it's a creation of a human mind, intellect. It is intangible property, and it gives a exclusive rights. Of course, it is attended with limitations and exceptions. It will be time bound, and it will have a territorial boundary. So what is intellectual property? Intellectual property is a property that arises from the human intellect. It is a product of human creation. It will be produced in two different forms. First one is a literary and artistic work, and the other one is industrial property. In case of literary and artistic work, these are the examples like books, paintings, musical composition, plays, movies, radio, TV programs. All these are protected by copyright. And in case of industrial property, because industrial property describes physical matter that is the product of an idea and concept of commercial purposes. This can be protected by using the most important is patent, and the other are trademarks, industrial designs, trade secrets where patent is not applicable, and the layout designs. There are three major types of IP, that is patent, copyright, and trademark. If it is a functional and technical invention, it will be protected by using patents. If it is a purely artistic work, it will be protected by copyright. If it is a symbol, logo, word, sound, color, design, it will be protected by using trademark. Apart from this, there are different other intellectual properties. They are classified under industrial properties like industrial designs, GI, geographical indications, layout designs, trade secrets, and the new one is a protection of new plant varieties. Now you'll see one by one in brief. The first one is copyright. The copyright law protects the expression of an idea, not the idea itself. Like patent, there is no requirement of novelty and uniqueness for having copyright registered. It should be an original work originated from the author. So literary, films, dramatic and musical, artistic and sound recordings, these kind of works are covered by copyright. And the ideas, facts, recipes, and work lacking originality, this cannot be protected using copyright. Ideas can be protected using patents. The scope of copyright is like that. It is a original literary, dramatic, musical, and artistic work, cinematographic, films, sound recordings, and it gives two different kinds of rights. One is moral right, and the other one is economic right. The economic right is to bring economic benefits to the author. If it is a literary work, it will be protected. The ownership of the right will goes to author. If it is a drama, it will go to dramatist. If it is a music, the right will go to composer. If it is artistic work, it will go to artist. Similarly, like sound recordings, it goes to producer. The films, it goes to producer. Photograph, it goes to photographer. The normal duration of copyright is like a lifetime of an individual plus 60 years. In case of some exceptional things like photographs, cinema, and recordings, it will be for 60 years from the date of publication. Now we will see trademarks. The trademark means something like a representation, a style of representing something. So we can see these examples like this Lux, Godrej, TVS, all having a different style, even though all these are names, but they are represented in a different fashion, different style. So these are the trademarks for that particular product, that particular company. The trademarks can be sign, words, letters, numbers. It can be a drawing, picture, emblem, color or combination of colors, shape of goods, graphic representation or packaging, or any combination of the above. So how trademark will be applicable? A trademark is a sign used on or in connection with the marketing of a good or services. Used on means it will be, the trademark will be written on the wrapper as well as the container and on the product. Like all other intellectual properties, trademark is also registered by national trademark registers and valid in that particular country. It also has a territorial, it is a territorial right. So it will be valid only within that country where it is registered. Registration is made after examination and publication. Period of registration is 10 years and it can be renewed indefinitely. There are different kinds of trademarks like marks on goods, service marks, certification trademark. There are different types: collective marks, well-known marks, trade names. We'll see a few examples. For example, this Coca-Cola. So it is a well-known mark. It will be represented using this particular style, the way it is written Coca-Cola. And this triangular shaped chocolate Toblerone will be represented by this way. A unique style of writing the name, chocolate name. And we often see Godrej products means on furniture, refrigerators, storewell, and many other Godrej products. It will be, we can see this kind of the name written in this particular style. This is trademark of Godrej. Hello? Am I audible?
Yes, sir, you are.
Okay. And the next example is GE. So this GE is represented in this particular style. It's not written as a simple G. It is represented in this particular style. And so this has become a trademark for that particular company. There are different symbols, like if it is if the trademark is filed for intent to use application file for product, then it will be written as TM. If the intent to use application file for services, if it is service related, it will be SM. And once the trademark is getting registered, the R will be written like this style. If this symbol is there, then it is a registered trademark. And this is the registration procedure. So first we need to apply for application for search, then application for registration, then the examination of trademark happens, then advertisement of trademark. If somebody want to oppose this trademark, he can oppose by filing an opposition. And if nobody file opposition and the trademark office is convinced with the uniqueness of this trademark, then they issue a certificate, and that way the trademark is getting registered. And now the topic of your interest, because we will be covering this IPR in pharmacy current scenario. So this will be a topic of your interest. So I'd be able to explain this only by your participation. So to understand and discuss this topic, your participation is desired. So what is patent? As everybody knows, it is an exclusive right to stop others from practicing your invention. And often it gives a technical solution to a technical problem. It is a limited monopoly rights. It is valid only for 20 years. And it gives you several rights to stop others from manufacturing, using, or selling the patented product without your permission. So in short, what is a patent? So it is a statutory right to the inventor or the applicant by the government for his invention, which is either a new process or a product. It is kind of an agreement between a inventor and government. So you have come up with a new product and new process, and the government is convinced with your invention, and they are giving you a patent to have a monopoly right for 20 years. It is for the limited period of time, 20 years. It is granted in lieu of sufficient disclosure to the patent offices. You need to disclose your invention in a sufficient manner so that you can obtain a patent. It is only a territorial right, means if we have filed a patent application in India, and if we own a patent in India, it will be applicable only in India. It will not be applicable to US, Europe, or any other country, unless and until we will not file a patent in US and Europe or any other country. It is a negative right. It is to stop others. Even though it gives some exclusive rights, there are certain exceptions, like for experimental use or government use. For research purpose, you can practice the invention even though it is patented. And in case of like this kind of pandemic, the government may intervene, and they will ask your permission to use the patented invention. So what are the important of patents? It is important source of scientific and technical literature. You can have a lot of science sitting in the patents. It is a treasure house of scientific inventions, correct? And it avoids duplication. If you know that somebody has done this particular work, you don't have to repeat that same work. So you can find out that somebody has done this work. I should not repeat this. So it avoids duplication. It paves way for further discoveries, and you don't have to reinvent the wheel. It stops reinventing the wheel because of the disclosure of the scientific disclosure in the patents. It identifies new areas, new technologies, emerging areas, what we should do, what are the new requirements in the pharmaceutical sector or any other sector. So patent gives you 20 years of right. So within those 20 years, no one else can manufacture the product. And if somebody want to practice your invention, he has to take a license. He has to license the patent from you, and he has to pay you royalties. And you can block the competitors. So no competitor will be able to practice your invention within those 20 years. And after 20 years, technology goes to public domain, and it becomes like a normal publication. After 20 years, anybody can use your invention. So what are the patent rights? Hello? Hello.
Yes, sir.
Yes, sir.
Okay, okay. So I can see that window here. So that is why I thought that it is some issues there. Okay, I'll continue. What are patent rights? It is to prevent third party without authorizations from making or manufacturing, using, offering for sale. You cannot even offer for sale. You cannot sell the patented product. You cannot import it from any other country. You cannot distribute it. You cannot license it to any other person. So these are the rights given to the patent owner. And if somebody want to use the patented invention, he has to take a legal permission from the patent owner. And in that case, he may have to pay the royalties. What are the patented subject matter? This kind of article, apparatus, or machinery on its component can be patentable. In case of pharmaceutical, these substances like new chemical entities or even the living or non-living products, pharmaceutical products, like formulation, new combination patents, new drug combination patents can be patented. Or any process or manufacturing of different style of manufacturing of biological process is also patented. So under Indian Patent Act, Section 2(1)(j), invention is defined as invention means a new product or a process involving an inventive step and capable of industrial application. Now I'll explain what is inventive step and industrial application. First, these are the basic criteria of patentability. To obtain a patent, you must fulfill this criteria. And the first one is novelty. Your invention has to be novel, new, unique, and it should not be. Hello. It should not be obvious to a person's skill in the art. It should be non-obvious. Means it should have some inventive merit. And the third one is industrial application. It can be easily practiced by in the industry or any other place wherever it is required. Then only it fulfills the criteria of industrial application. So what are patentable subject matter? So invention must be relates to process or a product or both. A product means like a new chemical entity. In case of pharmaceutical, it can be a new chemical entity, a new molecule, new chemical entity. And it can be a formulation product, a new unique formulation product. It can be a combination of two known product. That will also fall into product category. And the process means like a process to synthesize particular molecule, a new molecule, old molecule, anything, a new process, a new route of synthesis, a new way of formulating something. So that can be covered under this process. It has to be novel, be new. It should involve an inventive step. It should be capable of industrial application, and it should not fall under Section 3 and 4. See, Indian Patent Act says that under there are listed several inventions that are not patentable in this Section 3 and Section 4. I will explain that. First, we'll see what is mean by new. It should not be published in India or any other place globally. It should not be published anywhere in prior public knowledge or prior public use within India. It should not be there, and it should not be part of any specification claimed before we file the patent application. Inventive step means a feature of an invention that involves technical advance as compared to the existing knowledge. So technical advance as compared to the existing knowledge means something is already there in the public domain, and if we can improve on that, so it will have an inventive step. That way it will fulfill the inventive step criteria. If it has the economic significance, it is very good. So again, it fulfills the criteria. And it should not be obvious to a person skilled in the art. It should be non-obvious. As I explained, industrial application means invention is capable of being made or used in any kind of industry. Again, what can be patented? So the product or processes, if and only if they can fulfill this criteria of novelty, non-obviousness, and industrial application. There are mainly two categories of patentable inventions. In case of pharmaceutical products, the products means a new chemical entity, a formulation, new formulation product, a combination of two drugs with some synergistic effect will fall in this products category. And in case of process, pharmaceutical process, a new synthetic process of an old molecule or even new molecule will be covered in this process, as well as a new way of formulating some known formulation that will be covered, protected using this by process patent. So in India, the grant of patent happens in this way. Patents are granted by national patent offices after publication and examination. In India, there is a provision of pre-grant and post-grant opposition. If somebody feels that your invention is not novel, not inventive, or it is lacking some patentability criteria, he can file a pre-grant opposition in Indian Patent Office, and then it will be examined by the controllers on the merit. And similarly, even after getting a grant of a patent, within 1 year's time, any other interested party can file a post-grant opposition by citing novelty, industrial application, and inventive step, lack of inventive step, lack of novelty. They can file opposition. They are valid within the territory limits. Even in case of India, even foreigners can also apply for patents. But for that, they need to have somebody, some representative in India. They can hire a patent agent and they can file. Even the foreigners, the US, Europe citizens can also file a patent in India with the help of some representative, especially the patent agents, patent attorneys. So what is not patentable as per Indian patent law? Like inventions falling within the scope of section 1 subsection 20 of Atomic Energy Act, like inventions related to compounds of uranium, beryllium, because this is all related to the security of country. So these kind of inventions are not patentable. As such, nobody can have a right to stop anyone by having a patent on this. So that is why it is not patentable. And there are, under section 3, there are certain patents, certain inventions that are not patentable. The first one is like if an invention is a frivolous or which claim anything obviously contrary to the well-established natural laws. For example, a machine giving more than 100% performance. We know that it is not possible. So these kind of inventions are not patentable because it falls under the frivolous category. And the next one is like anything like kind of, see, for example, the gambling machine, the device for housebreaking, all these are not patentable because its intended use or commercial exploitation of which could be contrary to public order or mortality. So that is why it is not patentable. And this is very important. It comes under section 3D. Not this. This is important, but this is not a section 3D. It is a mere discovery of a scientific principle. Formulation of an abstract theory is not patentable, because as we know that discovery adds to the human knowledge by disclosing something not seen before. But in case of inventions, it adds to human knowledge by suggesting an inaction. So invention is like a kind of a solution to a problem. It is giving you the way of doing something. So that is called as invention, a new way of doing something. It can be a product, it can be a process. So that is called as invention. Like this Archimedes principle, and all these are not patentable. This is very important when it comes to pharmaceutical patenting. This is section 3D. Under this section 3D, the mere discovery of a new form of a known substance which does not result in the enhancement of the known efficacy of that substance. Listen, if some a particular molecule is already patented in India, and if you want to prepare a polymorph or a new form of that patented molecule, old molecule, it cannot be patentable in a normal normal scenario. To have a patent, we need to show some enhancement of the efficacy, like it should be at least 30% more efficacious than the original molecule. Then only it can be patentable in India. Again, the second one is the mere discovery of any new property or a new use for a known substance. If a molecule is used for particular therapeutic area, suppose if a molecule is an anti-diabetic agent, and tomorrow we find that this molecule is also used for anti-cancer, then in that case, the same molecule cannot be patentable for any other use like anti-cancer. This I am talking especially about Indian patent scenario. Again, mere use of a known process. These things are not patentable, but there are certain exceptions. If the process is resulting into a new product or employs at least one new reactant. The process is using a new, in case of synthetic molecules, if it uses some new reactants, then that process becomes a new process, and in that case it will be patentable. And this is again very important when it comes to formulation or combination patent. This under section 3E of Indian Patent Act, a substance obtained by mere admixture resulting only in the aggregation of the properties of the components are not patentable. Like for example, paracetamol plus brufen. So whatever the drug will result, it will have an antipyretic activity, that is the paracetamol activity, and brufen, it is analgesic activity. If the new drug resulting with this combination only has antipyretic and analgesic activity, it is not patentable, because it is very obvious, it becomes very obvious. They independently has this activity, and after combination also they only has that activity. So that way it is not patentable. Similarly, like a soft drink that is only a mixture of sugar and some colorant in water, it is not patentable by Indian patent law. But if a mixture resulting in synergistic properties, then it is patentable, like soap, detergents, lubricants. And again, mere arrangement and rearrangement of a known devices is not patentable. Like we know that, for example, a bucket fitted with a torch. There is a torch, there is a bucket. If we are fitting that together and just rearranging the things, that is not patentable. Umbrella with a fan. This is related to farmers. Method of agriculture or horticulture, this is also not patentable. However, the equipments are patentable. Agriculture equipments are patentable. Plants and animals in whole or any part are not patentable. This is like a mathematical method or a business method, a computer program. This is again an outcome of a mental process only and do not involve industrial process or products. So this can also will fall into the non-patentable inventions. And there are certain few more like literary, dramatic, musical, and artistic work. As we have already seen that this can be only protected using copyright. It cannot be protected using patent. A mere scheme or rule or a method of performing mental act or method of playing game like a crossword puzzle, method of solving a crossword puzzle, method of learning a language, method of teaching or learning. These kind of things are not patentable. Again, a particular way of presenting information is also not patentable. A topography of integrated circuits, like for example, the traditional knowledge is also not patentable. The traditional knowledge can be patentable only if with the use of that traditional knowledge, if we can come up with a new process or a new product possessing novelty, inventive step, and industrial application, then it becomes patentable. And inventions related to atomic energy are not patentable because it is related to the safety and security of the country. So it is not patentable in India. So what are the different steps of obtaining a patent? So we will see. It's like first thing, who can file a patent application, where to file a patent application, and how to file a patent application. Who can file a patent application under section 6 and 134? The application can be filed either alone or jointly. It can be a single inventor or it can be a group of inventors. And the person has to be a true and first inventor if you are filing the application in person. And if you want to assign it to someone, then by the use of patent agents, patent attorneys, you can also file a patent application. And in case of deceased person, a legal representative of any deceased person or assignee can also file a patent. See, to file a patent application, there are two ways. One is a physical filing at the patent office. You have to visit the patent office and file a patent application. From 2007, the electronic filing is introduced. So nobody nowadays files a patent application physically, and government is giving 10% discount on the fees if we are filing electronically the patent application. And if you are a citizen of South India, like if you are a citizen of Andhra Pradesh, Karnataka, Kerala, Tamil Nadu, and Union Territories of Pondicherry and Lakshadweep, you need to file a patent application in the patent office branch of Chennai. And if you are a citizen of Maharashtra, Gujarat, Madhya Pradesh, Goa, Chhattisgarh, Daman, Diu, you need to file a patent application in Mumbai patent office. In case of North, like Haryana, Himachal Pradesh, Jammu, Kashmir, Punjab, Rajasthan, UP, Uttarachal, Delhi, all this, Chandigarh, even Chandigarh, we need to file a patent application in New Delhi patent office. And for all the rest of India, we need to file a patent application in Kolkata Patent Office. What are the documents required for filing a patent application? If you are filing physically, then covering letter indicating the list of documents, and you have to fill up certain forms like Form 1, application for grant of patent, and the next one is very important. That is Form 2, which has your actual patent application, the complete specification of your patent application. And then another important form is that Form 3, where you have to specify whether you have filed this patent application in any other countries other than India. That you need to mention in this Form 3. This is also mandatory. And if you are using any patent attorney or any other assignee or any patent agent or any other representative, then you have to fill up this Form 26, that you are authorizing some person for filing a patent application on behalf of you. And the Form 5 contains the declaration of inventorship. It will have the name of the inventors and it will their addresses. And Form 18, it is also very important. We need to file Form 18 for requesting examination. Unless and until we will not file Form 18, our patent application will not be examined. And there are this can be filed within 48 months from the date of filing of the provisional application. And we have to pay the requisite statutory fees. If it is physical filing, check or DD, or if it is a online filing, you can pay online. And this is the Form 1. So what kind of information it will have? It will have kind of a legal information. Will say the name, address of the applicant and the inventors, their contact details. If it is assigned to someone, then the assignee names, assignments. This is how the Form 1 looks. This is a mandatory requirement for filing a patent application. You Form 1 and submit along with your provisional or complete patent application. And this is important one. This is actually your patent application. So this is called as Form 2. So how it should be, means the Form 2 should have a background of the invention. It should describe the need of the invention and summary of the inventions, how the invention was. Someone should be able to duplicate your work, and the preferred embodiments like examples. And the most important portion is like claims. And the abstract of the patent application, the summary. As I explained, this is a Form 3 where you have to give all the foreign filing details. Declaration of inventorship, where you have to list all the inventors with their addresses. And if you want to have early publication of your patent application, you can file a request for early publication using Form 9. And this is a very important one. You need to file a request for examination for getting your patent application examined within 48 months. There's a Form 26. This will be required if you want to authorize someone to practice on behalf of you. Then you need to file this Form 26. And what are the essential ingredients of the complete specification? Like a publication, it will also have a title, a preamble, a prior art, discuss in detail, drawbacks in prior art and what kind of solutions we are proposing over the drawbacks of the prior art, and the summary of your invention, the statement of your invention, the detailed description of inventions. Means you have to explain your invention in detail by using some embodiments. And if you are using some drawings or something, you need to mention all those figures. And the most important one is examples. This is the experimental section. This is very important. This will be considered as your disclosure. So this is very important. And then the next one is claims. So whatever you are claiming and whatever claims are granted to you becomes your patent right. These are the main steps of patenting procedures. Like you need to file a patent application at the patent office. The next one is a request for examination, followed by it will be published after 18 months from the date of filing, initial date of filing. Then you have to fill up that form 18, and after that it will get examined. And then after that examination, the first examination report will be released. And before grant of a patent, anybody can file a pre-grant opposition. And if the controller is satisfied with your submissions, the controller will grant you a patent. And within a 1 year of time, any interested party can file a post-grant opposition. Only within 1 year. And this is the way the patent granting system is in India. First thing is that very important, conceptualizing an invention. Then the next one is you need to file a patent application. It can be a provisional application. If you are filing a provisional application within 12 months, you need to file a complete specification. And if you are ready with your invention, you can directly file a complete specification. After 18 months, it will be published. And if you are failing to file a complete specification within 12 months, your patent application will get abandoned. And pre-grant opposition is also possible. Request for examination is required. And once the request for examination is done, the examiner, the controller will examine your patent for patentability criteria like novelty, inventive stuff, and industrial application. And then he will issue a first examination report. And we need to answer to his queries, his questions in that our reply to the first examination report. Then he will re-examine the amended documents, and if required, he will offer you a hearing. Then you need to explain your invention. It will be a kind of a mutual. It will be a kind of a one-to-one discussion with the controller, the examiner. Then you will be able to explain and convince him about your invention. And in general, the application has to be put in order within 12 months from the issue of first examination report. And if objections are not made within 12 months, it will be abandoned. If objections are made within 12 months, it will be granted as a patent under Section 43 of Indian Patent Act. And within 1 year, anybody can file a post-grant opposition under Section 25(2). So if you want to ask any questions related to patents, we can discuss now, or otherwise I'll continue and I'll conclude the presentation. Then you can ask the questions. Anyway.
Sir, yes. Good afternoon, sir. Dr. Lavati.
Okay, please.
Yes, sir. You have given really a good insight on the patents, and that is a part of your the syllabus of the final year, also the MPharm students.
Okay, very nice.
You definitely add to their knowledge. Just one query I want to ask, because as per the Indian patent law, we cannot go for the patenting of new use of any particular substance or API.
Correct, correct.
Yeah. But in US, it is patentable, sir?
US it is patentable. US it is patentable. Europe also in a different way it is patentable. US it is patentable, 100% patentable.
Because I've just gone through one patent yesterday itself of US patent, and they just find out the new use of their I think leocetrizine molecule,
Yeah,
and
it is.
they patented that. So
Okay,
actually, we are also certain projects in our mind. So can we patent in India also in
No.
other way,
India,
some of the other way?
yeah, India does not allow that provision because our purpose of patenting is like we want to prevent the evergreening of patent. We don't want to give any monopoly right to anyone for larger number of years.
Okay.
So we want to restrict
Because
only
evergreening strategies there now.
Hello.
So can we suppose we prepare any particular formulation and then we have to go for patenting. So formulation will be patented or the use will be patented, sir?
The formulation you can patent, but if you want to use that formulation for particular use, it will not be patentable, but still you can use that by applying to DCGI and everything. You can use that particular formulation, but you will not be able to obtain a patent for using that formulation for that particular use. You may get a patent for a formulation. Since it is a new formulation, you can get a patent, but you will not be able to get a patent for a particular new use or new use.
New use we cannot be patented.
It cannot be patentable in India.
Yeah, and sir, second question is there in the what is the criteria for patentability of co-crystals?
Co-crystals, yeah, it is again it is kind of patentable one way. Like right now we are seeing a lot of all these anti-diabetic molecules. All these new molecules, they are coming up with co-crystals. So it is that way patentable, but still you have to prove that it is your invention satisfies the criteria of novelty. Novelty obviously it satisfies, but the inventive step means how it is inventive. If you are preparing a co-crystal, how it is beneficial over the normal product.
We suppose already the molecule is patented and we have developed a new co-crystals of that particular molecule. And so we have to prove the more efficacy, maybe a dissolution or maybe solubility or maybe the bioavailability, that we have to prove.
That way means it will not result into a product patency. It will be kind of a particular formulation patent. Correct? It will not be considered like a new molecule. It will not be considered as a new form of a known substance. If a substance is known, we are eligible to patent it only once. And for this co-crystal, it will be kind of considered as a formulation patent. And if it satisfies the criteria of non-obviousness, it can still be patentable.
Because it will be, it is not a formulation, sir, just a modified form of that particular entity.
Yeah, I fully understand your question. It is co-crystal, correct? Yeah. So as
and
such,
in co-crystal, actually what we have done, we have developed the drug co-crystals.
Yeah.
Drug theoretically, two drugs, they are combined together.
Okay. So theoretically, it is patentable, but you need to prove that what kind of advantages you are getting by using this co-crystal drug combination over the standalone single drug and the second drug, first drug and second drug. So if you are able to prove that it enhances your stability, the impurity profile is getting reduced. So that way it becomes a patentable subject matter, but it will not be considered as a kind of a new chemical entity. It will be a kind of a combination formulation product, either combination product or a formulation product. It will be considered as a combination product. It is that way patentable, but you need to fulfill this criteria. Like you have to show all those non-obviousness feature of that.
Thank you, sir.
With the synergistic effect has to be seen. Then only it will be patentable.
Synergistic. Safety and efficacy has to be proved then again.
Safety and efficacy, if it is, safety and efficacy, if it is considered as a new product, then yes, you have to prove the safety and efficacy. If it is not approved product in India, then you have to prove the safety and efficacy. You have to approach DCGI for getting approval for your new product. Then safety and efficacy is required.
Thank you very much, sir, for your. Thank you, sir. Thank you, sir. That's on my side.
Okay, okay. Any other question?
Sir, Mangesh sir.
Yes, please.
Sir, is there any specific format to file the reply to first examination report?
For you're asking about some format.
Format, yes, sir.
Okay, means there is no specific format, but means like you have to there will be see the examination will be conducted for checking whether your invention is novel, whether your invention is inventive, or whether it satisfies the industrial application criteria. So the controller will the examiner will have all these objections in your first examination report, and whatever the objections are there in the first examination report, accordingly you need to prepare your reply. If it is a novelty related objection, you have to explain how it is novel. If it is related to inventive step, then you have to explain how it is inventive over the other prior arts, and mostly industrial application will not be there because it is a pharmaceutical product. So these two are the main things. And as apart from that, under section 3, there will be a lot of objections to the pharmaceutical products. The section 3E especially under that it will be objected for since it will be if it is a combination of two products, then they will say that you show the synergistic effect, then only it will be patentable. So this kind of objections will be there, that accordingly you have to reply. Means that way there is no specific format, there is no template as such, but you have to see how it is objected, and you have to reply according to that objection. And there will be certain like non-technical objections also, where you have to reply accordingly.
Okay, sir. Okay, sir. Thank you, sir.
Okay. Any other question?
Sir, my question is related with law.
Related to?
Or we can say that the trademark of a company. Yeah.
Okay.
So, for example, some person copies the logo of another company. Okay, and he has just changed the background color. Suppose, say for the example of Coca-Cola, the background color is red, and he has changed the color to some other color. So, can he file the trademark, or is it a case of infringement?
It will be case of infringement because it becomes very obvious.
Okay.
Correct. It becomes very obvious that anybody can change that color of the background color and file a trademark. So it becomes very obvious.
Okay, thanks.
Any other question, please?
Any questions from the student side? Students, you can ask your valuable questions.
Yeah, please don't hesitate. Means you can ask any question.
सर पेटेंट को अप्लाई करने के लिए कम से कम क्वालिफिकेशन कितनी जरूरी सर? लाइक बीकॉम थर्ड ईयर और फाइनल ईयर स्टूडेंट अप्लाई कर सकता है क्या सर पेटेंट के लिए?
हाँ, ये कर सकता है, जरूर कर सकता है.
ओके सर, थैंक यू सर.
Any other questions?
You can ask any innocent question. No issue that way. Students.
And from students, research students, do ask your questions. So, okay, so it seems no questions will be there.
Okay.
So thank you so much, sir, for this excellent session on intellectual property rights. After completion of this session, we came to know many examples related with patentable and non-patentable drugs or examples.
I think sir is covering geography in GI also, no?
Yeah.
Yeah, also I'll cover. Shall I continue?
Okay, sir. Okay, no problems.
Okay, yeah. So we'll see GI. So as the name itself indicates, it is related to something, a product related to a particular geographic origin. So for example, this Nagpur orange, it is Nagpur. It is related to Nagpur city. If it is, this is the second one is the Paris perfume. It is related to a particular city of France. And Banaras silk. The silk sarees are related to this Banaras silk. So it is a geographic, these products are related to a geographical origin. That is, these are protected under this geographical indication. This is the definition. A geographical indication is an indication which identifies goods as agricultural good, natural goods, or manufactured goods as originating or manufactured in the territory of country or a region or a locality in that territory, where a given quality, reputation, or other characteristic of such goods is essentially attributable to its geographical origin. And there are certain exceptions like Alfonso and Basmati. It's not related to a geographical name, but still they are protected under geographical indication. These are certain exceptions. And the goods can also include the goods of handicraft or industry and food stuff. Like patent or trademark, it will also require some registration. It will be examined and published, and then it will be registered. The registration will be valid for 10 years and it can be renewed indefinitely. Like all other intellectual property, it also gives you certain rights. It gives the exclusive right to use the geographical indication on the goods. And in case of infringement, we are able to claim the relief. And now the other one is trade secrets. So we often use this word trade secret. See, something what is not be protected by using any of this intellectual property like patent, copyright, trademark, then this can be protected using trade secrets by keeping it confidential, by keeping it not known to most of the people working around the projects, or in case of so the recipes, we often see that there are certain recipes, there are certain in Aurangabad also we can see there are many like the biryanis and other shops, the sweet shops, they will have kind of a, from the generations they are selling that particular product, and so that way they are maintaining their trade secret. So it will be mostly related to if the invention is not patentable. And see, the patent gives only 20 years of right. If it is a trade secret, it can go by generations that way. So the cost of patent is a little high. So to prohibit that cost, so we can have a trade secret. When we know that it is difficult to reverse engineer, then also we can go for a trade secret. How can we guard a trade secret? We can restrict the number of people having access to secret information, signing a confidentiality agreements like a CDA, non-disclosure agreements with business partners as well as employees. There are certain protective techniques like digital data security tools and restricting entry into area where trade secrets were or held. And many national legislation provide protection in form of injection and damages if secret information is illegally acquired or used. I'll see industrial design. Means design is something like a particular shape, a configuration, a particular style, a shape mostly. It will be shape, some design. These are few examples, like, so we can see this Kolhapuri chappal, this brush, this pen, this watch. It will have a particular design. So this will be protected under industrial design. In case of pharmaceutical products, these ampoules, these capsules, these inhalers, these syringes, all these these vials, these are having a specific design, and this is protected using this industrial design. And industrial design is very popular in textile and jewelry industries. So we can see this this cloths where they will have a different style of different designs. The flowers or any other designs will be there, and the jewelries, it will be it will have a particular shape. So this is how it will be protected under industrial design. And with this, I'll conclude. Thank you.
So, okay, thank you, sir. After completion of this lecture, finally we got the knowledge in the step-by-step process of filing the patent. So
Yeah.
we are very thankful to you, sir. For this lecture.
Okay.
Thank you, sir.
Welcome. You can still ask some more questions.
Yeah, definitely. Is there any questions?
It can be any innocent question, as I said earlier, because students may not know all the things, but still they can ask some question.
B.Pharm final year students, M.Pharm students, you can ask your questions. So it seems that no questions will be there.
Okay.
So once again, thank you, sir. Thank you so much for your lecture. Thank you, sir.
Okay. Thank you.
So with this, there is one announcement that the feedback link is there in your chat box. So kindly fill that link, get your certificates. And one more thing is that we are looking forward for the second session of this Impact Lecture Series 2. That will be probably after 10 days. So don't leave this WhatsApp group. Okay. So on behalf of IB Chavan College of Pharmacy, Institutions Innovation Council, I would like to express my gratitude to all esteemed delegates of the webinar for their presence and contribution to make this webinar a great success. I extend my gratitude to our management principal, Dr. Abu Bakar Bawazir sir, Vice Principal Dr. Santosh Mokle sir, IQAC in charge Dr. Degan sir, NIRF Coordinator Dr. Lahoti sir, IPR Activity in charge Dr. Sangshetty sir, ICT committee members Dr. Ismail Mozam and Mr. Altamash Ansari sir, and all the staff members to make this event a grand success. So thank you so much.
Thank you, sir, for your such a nice lecture, sir.
Okay, thank you. Welcome.
Thank you, sir.
So we can leave